Hello guys, welcome back to another video. Today we're going to be going over vaccines, how they work, and the concept of herd immunity, because apparently some people still don't believe in them. Expect better pronunciation in this video due to the lack of Aztec gods. So, time for some context. Vaccines are based on a practice known as inoculation, which is artificially transmitting a pathogen to an organism in the hopes of the organism becoming immune to it. However, the first vaccine was created by English physician Edward Jenner. He also coined the term itself. Before we get into how vaccines actually work, let's talk about the process it takes to make them. Because let me tell you, the bureaucratic nonsense to actually get these things into use is probably deserving of an award in of itself. The first step of vaccine development is the exploratory or discovery phase. So, in order to first create a vaccine, they have to target a pathogen. I know, shocking, right? After they select the pathogen, they conduct in depth research on how it functions. They investigate how the pathogen infects host cells, replicates, and evades the immune system. Next, scientists must identify the mechanisms the human body uses to fight off that particular pathogen. This is usually done in the form of antigens. Researchers identify the most appropriate antigens that can induce a strong and protective immune response against the pathogen by the creation of antibodies that match with the antigen to fight it. After they have decided what antigens they want to target, they need to begin trials with in vivo and in vitro testing. In vivo testing is testing on live organisms but not humans. In vitro testing is testing done within a culture, laboratory dish, or test tube. It is in this stage that researchers use testing to find the optimal dosage to enhance its stability, efficacy, and safety. This testing is done on a small scale and mainly done for proof of concept. After reviewing all the data they collected in the exploratory stage and submitting it for review, researchers are ready for the next step in vaccine development, preclinical trials. This stage involves mass animal testing on a variety of animals under various laboratory and clinical conditions. This is mainly done to test the effectiveness of the vaccine and ensure that there are no unexpected side effects. After the preclinical trial stage, it's time for the clinical trial stage. Clinical trials are primarily divided into three separate stages. The first stage focuses on testing in a small group of healthy human individuals in order to see the effects on humans. If the results from the first stage are promising, then the second stage involves testing of a larger group of people who have certain medical conditions or characteristics to represent the target population for the vaccine. While children are sometimes included in the second stage, they're usually not tested until stage 3. Finally, in the third stage, the vaccine is tested on a much larger and diverse group of people, usually in the thousands, using scale to further evaluate its safety and effectiveness. This stage also helps identify any rare side effects that may not have been included in the smaller groups. This now includes groups that may be at risk by taking the vaccine, such as children or elderly people. Once the clinical trials are successfully completed, the vaccine manufacturer submits all the data to health regulatory authorities for review and approval. These authorities assess the data and determine whether the vaccine is safe for public use. In the case of a positive result, the vaccine is given the green light and given the license to market, which means the vaccine can be included in public health systems in that country. But we're not done with the vaccine process yet. After the evaluation and decision phase, we enter the manufacturing and safety monitoring phases. These phases occur simultaneously. The manufacturing phase is when the company or IP holder of the vaccine begins and expands mass production of the vaccine. Over time, the process produces more vaccines at a more efficient rate. The safety monitoring phase is the continuous recording and rigorous testing of the effects of the vaccine, now with the data pool of everyone the vaccine is being used on. Should a vaccine be proven to be harmful after the safety monitoring phase, it can be recalled back. However, the primary changes that happen due to the safety monitoring phase is changing the dosage and the interval that the doses are given in. So, as you can see from the list of steps in the vaccine development process, it can take a while, anywhere from 8 to 18 years, usually averaging around 10 to 15. However, some issues such as COVID-19 require a solution within a short time frame, and thus most governments have a separate set of regulations for emergency development. These regulations all often involve laxer regulations, less trials, and a quicker review process, which can lead to safety issues with these new vaccines. Now, let's get into the nitty gritty of how vaccines work. As I mentioned earlier, vaccines contain weakened or inactive parts of a pathogen, like virus or bacteria. These components are enough to trigger an immune response, but not enough to cause the disease itself. When we receive a vaccine, our immune system recognizes these harmless pathogens as parts of foreign invaders and launches an immune response to fight them off. This process involves the production of antibodies that specifically target the antigens found in the vaccine. Once the immune response has been triggered, the body creates memory cells called T cells that remember these pathogens' antigens. These memory cells will remain in our system ready to spring into action if we encounter the real dangerous pathogen in the future. This memory response is the key to why vaccines are actually effective. If we are exposed to the actual pathogen after vaccination, our immune system can mount a rapid and strong defense, preventing the disease from taking hold and significantly reducing its severity. The actual cells that are responsible for the memory in our immune system are called T cells. Now, let's talk about herd immunity. Herd immunity occurs when a significant portion of the population is immune to a particular infectious disease, either through vaccination or previous infection. When a large portion of the population is immune, the spread of the pathogen becomes limited. This indirectly protects individuals who cannot be vaccinated or are more vulnerable to the disease, such as those with medical conditions. Herd immunity is essential, not just for individual protection, but also for the general public health. It helps prevent outbreaks and keeps infectious diseases under control. But seriously, I know that the video so far has been lighthearted, but if you believe that vaccines can cause harm to you, either through microchips or causing autism, I implore you to reconsider. Giving all US citizens microchips would be a logistical nightmare and cost more than NASA's budget, as well as constant replacements. Autism, on the other hand, is caused by genetics, environmental chemicals, improper brain development, and even some viruses and infections. So, vaccines actually lower your chances of getting autism. Not getting vaccinated not only negatively impacts your health, but can affect the most vulnerable people in society: children, the elderly, and people with life threatening illnesses. On that happy note, that's it for this video. I hope you have a great day and be sure to like this video and subscribe for more content.